This is a quick revision video on DNA, genes and chromosomes in the genetic information, variation and relationships between organisms topic in AS biology. Just worth a note, this revision video is AQA specific, and if you're doing OCR, Edexcel or WCAG, I think the Welsh one, then some of this content won't be specific to you, but you can still watch it if you feel like it'd be helpful. Okay, so let's go. This first part of the topic has a ton of definitions in it, which is a pain in the butt, but we have to learn them, so might as well. The genetic code is degenerate, universal and non-overlapping. It's degenerate because some of the amino acids have more than one coding triplet. I say some because not all of them do, some of them only have one. It's worth putting in just in case the examiner tries to pick you up on it. Universal because the same codons code for the same amino acids in all organisms, and it's non-overlapping because each base in the sequence is part of only one codon. Only two more definitions and then we're done. We have to know that the genome is the complete set of genes in a cell, and the proteome is the full range of proteins one cell can produce. We also have to know how DNA in prokaryotic and eukaryotic cells is structurally different. So in the prokaryotic cell, the DNA is short, circular and not associated with the protein. In the eukaryotic cell, it's long, linear and is associated with the protein. Also worth noting, chloroplasts and mitochondria, their DNA is structurally very similar to the prokaryotic cell. It's also short, circular and not associated with the protein. These are my dodgy attempts at a chloroplast and a mitochondria, which looks a lot like a hot dog. We know that eukaryotic DNA is associated with the protein, and that protein is called a histone. It needs to be associated with the protein because the DNA molecule is really long and has to be wound up to fit in the nucleus. It's wound around the histones, which are these little purple proteins here, and they help support the DNA molecule. The DNA molecule and the protein together is then coiled up tightly to form a compact chromosome. Worth noting that prokaryotic DNA isn't wound around histones because it's not associated with the protein and it's condensed by supercoiling instead. A gene is a sequence of DNA bases that code for a polypeptide or functional RNA. Examples of this are ribosomal RNA and tRNA, which is transport RNA. The order of bases in a gene determines the order of amino acids in a polypeptide, exactly like when we did proteins back in biological molecules. A triplet is a sequence of 3 bases, and a coding triplet is called a codon. There are 20 amino acids and 64 possible triplet combinations, which is why this code is degenerate. DNA is useless. Well, most of it is anyway. That's because a lot of DNA is actually made up of introns, and they are sections of non-coding DNA within a gene. We don't really know what they're for. Exons are coding sections of DNA within a gene, and they code for amino acids or functional RNA, and introns are removed during protein synthesis so they don't affect the amino acid order and furthermore the protein. Non-coding repeats are DNA sequences that repeat multiple times and don't code for anything, so pretty much does what it says on the can. Last but not least, alleles. As humans, we have 23 pairs of homologous chromosomes, which means chromosomes that are pretty much the same. Homologous chromosomes contain the same sequence of genes, but may have different alleles, and alleles are different forms of the same gene. The fixed position of a gene or an allele on the chromosome is called its locus. This diagram here just shows this all quite nicely. It shows that here we have 2 different alleles coding for 2 different things, one for purple flowers and one for white flowers. Now, although they're coding for different things, this is a pair of homologous chromosomes because the locus is at the same point and they are actually the same gene. They're just coding for different things. That's it for this video. Hope it helped you, and if it didn't, meh.